Hello, I'm Steve Weberg. I'm with the public affairs staff of the Kansas City Public Library, and I want to thank you for joining us tonight. While it's great to have you here virtually, we wish, just like we've wished for the past 15 months, that we were doing this in person, though I'm happy to say we're getting there. Library reopened its doors at the beginning of this month on June 1st, and while we're still not having large inside gatherings, we're hoping to return to in-person programming in our auditoriums this fall. And I can almost guarantee that we will look to reconnect with our guest tonight, Richard Preston, hosting here in Kansas City at some point in the future. We're rooting for another book. Almost 2 years ago, when Richard released what I think was the last of his 10 books to date, Crisis in the Red Zone, the story of the deadliest Ebola outbreak in history and of the outbreaks to come, he sounded a warning in its preface. With Ebola, which emerged in 2014, he said the world had seen the, quote, most destructive, rapid epidemic in anyone's lifetime, unquote. And he signaled that it, quote, may be an example of things to come, unquote. Now, Richard Preston is one of the most informed, most distinguished science writers of our time, of any time. An asteroid is named after him. And after 600,000 deaths in the US and closing in on 4 million worldwide from COVID-19, we know that he was all too right. There's no one better, and I respectfully include epidemiologists, immunologists, and scientists, to put into context what we've experienced the past 15 months, why we experienced it, including its origin, be it natural or from a lab, and what might lie ahead. Richard, as you might know, changed the way the world looks at pandemics with his first book about the Ebola outbreak, the 1994 bestseller, The Hot Zone. American Scientist magazine said it was one of the 100 or so books that shaped a century of science, with, among others, Albert Einstein's The Meaning of Relativity, the autobiography of Charles Darwin, works by Pavlov, Stephen Hawking, Carl Sagan, among others. Hollywood also based the 1995 film Outbreak on it, based it loosely, and National Geographic adapted the book into a 6-part scripted series. Richard, who is a prized contributor to the New Yorker, is renowned for the depth and detail of his reporting. And as a former reporter myself, I have great, great admiration for that and a bit of envy. And not just the depth and the detail, but the lengths to which he'll go to do it. He climbed California's giant redwoods himself in writing and researching The Wild Trees, his book, in 2007. He explored the Kenema cave near an extinct volcano on the border of Kenya and Uganda, where Ebola may have developed in bats. Among other honors, he's the recipient of the Science Communication Award from the American Institute of Physics. He's the only non-physician to receive the Champion of Prevention Award from the Centers for Disease Control and Prevention, the CDC. And then there's the asteroid, discovered in 1985, which travels primarily near the orbits of Mars and Jupiter, named 3792 Preston, or 3792 Preston. As Richard joins us, if you have a question for him over the course of the evening, you can submit it via the YouTube live chat box. We would invite you to do so, and we'll get as many answered as we can at the end of this discussion. Richard, I've looked forward to tonight since we first connected with you. Welcome, and thank you so much for being here.
Thank you. Thank you, Steve. It's really a delight and an honor to be speaking to the Kansas City Public Library. It is a great institution in its own right. I also really miss being physically in Kansas City. I wish I was there with you all right now. I love Kansas City. I could be going out and getting some barbecue and enjoying the place. At any rate, I'm going to be showing some slides tonight, and I'm going to be talking a bit about pandemics, about viruses, about Ebola virus, and also about the COVID pandemic and the interesting, profoundly interesting question as to whether or not this tremendous pandemic began as a natural event or as a laboratory accident. I'll be trying to tackle that interesting question. So, first of all, just to get to the basics, what is a virus? Well, here you see a cell, and growing out of that cell are hundreds and hundreds of particles of Ebola virus. A virus is a parasite. It's a very small particle that has the ability to replicate. The particle is typically consists of a protein shell. Sometimes it has a membrane around it. And then at the center of the particle, there is a small amount of genetic code, either DNA or RNA, which contains the blueprint and the recipe for the virus making copies of itself. Viruses are, as parasites, they can only make copies of themselves, replicate inside a living cell. The question of whether a virus is alive or not has never been answered and probably can't be answered. So biologists refer to viruses as life forms rather than as organisms, as living things. As life forms, viruses appear to be extremely ancient. They may have appeared early in the history of life on the planet. They even may be examples of the very first kinds of life on the planet. But we don't really know because viruses have not left any actual physical fossils in the fossil record. So we can't see them in the fossil record, but we can infer things about viruses by reading their code, their genetic code. But viruses are everywhere in nature. As far as we know, every kind of living thing has its own viruses that infect it, that replicate it, that use it as a host. Everything from bacteria to blue whales have viruses in them. But viruses as life forms can be thought of as being somewhat distinct from the biosphere, which is the collection of all living organisms on the planet. So we talk about the virusphere and we talk about the virosphere. The virosphere and the biosphere are dependent on each other, and they also are completely intermingled, like milk and tea or like mist in the air. But viruses are everywhere. They're absolutely everywhere. So if you take a liter of seawater, it will have more particles of virus in it than anything else, any other form of life. A liter of seawater has typically about 100 billion particles of virus particles drifting around in it. In the air we breathe, every cubic meter of air is thought to have about 10 million particles of virus drifting in it. So the biosphere is ancient for sure. It may be 4 billion years old or older, but the virosphere may be even older. It may have preceded the biosphere. In other words, the biosphere may have grown out of the virosphere. But viruses are everywhere. They're even inside our bodies, everywhere. The human body, it is thought that about something like 6,000 or more different species of bacteria inhabit the gut, our gut. And each one of those types of bacteria has its own particular viruses that infect it. But the viruses that exist in our gut and that are parasites of our gut bacteria are actually preserving our health because they keep the populations of gut bacteria under control and in balance, and therefore keep us healthy. if you were to eliminate all the viruses from the bacteria in our gut, we would almost certainly die, and quickly, of totally incurable dysentery. The gut bacteria would just go out of control. Populations would change completely. So one of the things that a virus can do, viruses have persisted for a very long time on the planet. They have their ways of survival and of perpetuating their code through time. And one of the more important features of a virus that has given it, as a form of life, an incredible survival capability is what is known as the cross-species jump. Viruses have a tendency to move from one type of host to another type of host. They tend to not be able to recognize perfectly the distinctions, the boundaries between species of hosts. And so, for example, dogs can catch colds from humans. Humans can catch colds from dogs. Typically, it's not a bad cold, it's just another cold. But as time goes by and as species of organisms appear and disappear in the long unrolling of deep time, viruses just simply move to new hosts. And as they move, as a virus does a cross-species jump, it changes, it evolves, and viruses can evolve quite rapidly at times, as we're seeing right now with the COVID pandemic, where we have alpha, beta, gamma, and delta versions of this virus emerging sequentially in different parts of the earth. Strains of the virus that are getting apparently better and better at dealing with and defeating the human immune system. This is rapid viral evolution. And so when a virus does a cross-species jump, it colonizes a new host, it changes, and it becomes intimately involved with the cells of its new host. So in terms of cross-species jumps, the human species has enormously increased in numbers in the last 120 years or so. And so we have become quite rapidly a very large, crowded, new potential host for viruses that are doing cross-species jumps, that are coming out of nature and moving into human bodies. My grandfather was born in the year 1900. And at that time, the Earth had a population of about 1.5 billion people. He lived a good long time. He passed away in 1998 at the age of 98, at which point the population of the Earth was more than 6 billion. So in one human lifetime, the population of the Earth had expanded dramatically. And the other thing that has happened is that for the first time in the history of the human species, more than 50% of people are living in crowded urban environments. When we were hunter-gatherers, we were very scattered and dispersed. And then since then, we have long been primarily agricultural. People have lived in villages and on farms, but more and more people are crowding themselves into cities. And what we're seeing is the growth of super cities. These are cities where the human population is 20 to 25 million people crowded into one urban area. And some among the largest on the earth are Greater Tokyo, with a population of something like 25 to 28 million. Greater New York City is one of the largest super cities on the planet. It has a population of about 20 million people living within one congealed urban area. Lagos, Nigeria, Dhaka, Bangladesh, Sao Paulo, Brazil, and so on and so forth. Now, if you consider some of these cities in the developing world, take about roughly the population of Texas, say 25 million people, and pack them into one city, put them really close together in slums, then take away access to medical care, take away sanitation, take away public health doctors, and then take away good government. And what you have is a ticking biological time bomb, a city that if a virus gets into it, it can go off like essentially like a bomb. And now the cities of the earth are linked together with airline routes, so that if one city gets a virus in it, all cities get viruses, the same virus, rather quickly. In 2019, the last year that airline travel was sort of normal, 4.5 billion people took a plane flight somewhere. Those were 4.5 billion passenger flights. And so you have two important factors going on here that make the human species exceptionally fragile right now. One is the packing factor. We're getting packed into cities. And then the second thing is the mixing factor. When you have the human population of the planet as mobile as it is, with people going all around the world very quickly, you have what amounts to a kind of essentially a blender that's set on high speed, that's putting every person biologically into contact with every other person on the planet pretty quickly. But from the point of view of a virus, the human species is nothing except a potential host, a great virgin territory to be explored, an enormous pile of meat. We don't, what we think, what our hopes are, what our dreams are, what our minds tell us are of no interest whatsoever to a virus. A virus is only one biological drive, which is to make copies of itself. So now we're passing through a really great pandemic, a pandemic that many people have unfortunately dreaded and expected would happen at some point. And the future arrived rather suddenly in December of 2019 when this coronavirus came, as it were, seemingly out of nowhere. What we know about COVID is that it very likely is its ancestral form was probably a coronavirus that lived in a horseshoe bat. There are lots of different kinds of horseshoe bats in Southwest China, and they have lots and lots of different coronaviruses. We think, no one really knows, but some scientists have done research. They've gone into these caves in Southwest China, and they've sampled the blood of bats, and they've been able to identify so far roughly 500 different coronaviruses that were previously unknown to science, but we think that they're like, nothing to a bat. They're basically bat colds. They probably give a bat a sniffle. They might not even make a bat very sick, but they have a tendency to make a cross-species jump into humans. Coronaviruses have been doing this for a long time, and for reasons that still are mysterious. This one particular coronavirus, which most likely began with a bat in a cave in Southwest China, somehow leaked into the human species and went on a burn. And now we've experienced 600,000 deaths. I think about this, and I think about exactly what happened here. And it is profound to think about. So the pandemic began with one human being, and that person has never been identified. We may never know who patient zero, the first case, the index case was, but somehow or other, a few particles of the COVID-19 virus leaked out of an ecosystem, may have come directly from a bat, or there may have been an intermediate host, another animal that the person was exposed to, but a few particles leaked into this one person. And the virus was able to set up a continuing chain of infection in humans, an explosive chain of infections. And within months, the world had an economic global heart attack. And here we see what happened in Times Square. Times Square was emptied out due to the shutdown. It's profound to think of. And it's also somewhat alarming and profound to think that it's not the first such plague, and it is certainly not the last either. You know, one thing that I've been meditating on is what are we really talking about when we talk about the COVID virus? What is this life form? What does it consist of? This thing is an enemy of our species. It is a parasite that has done incalculable damage and taken incredible numbers of lives and caused suffering beyond anybody's ability to really imagine it. But what is this thing? From this tiny handful of particles that got into one human being and then began to spread, how much COVID virus is out there right now replicating in us, in the human species? So I did a little calculation, and here's what I came up with. So particle of coronavirus looks like a golf ball with little fuzzy hairs all over it. And it is very small. So if you took 800 of these particles and you lined them up in a row, they would span the thickness of a human hair. If you made a layer of these particles, you painted that layer onto the head of a pin. The head of a pin could contain about 7 billion coronavirus particles. In other words, as many particles as there are humans on the earth. So how many people currently are actively sick with COVID? Well, I don't think anybody knows, but it's possible that the reported cases are a huge underestimate. We just really don't know. But just to throw a number out there, maybe a generous number would be about maybe 50 million people have coronavirus in them, and I calculated that would be around 1 quintillion particles of coronavirus. A quintillion is a 1 followed by 18 zeros. Well, that's a lot of particles. But if you took all those particles and you put them all together in one place, you would have a lump of coronavirus that would be not bigger than this potato, than this red potato. Might even be considerably smaller than that. So that's what we're talking about when we talk about the coronavirus that gave the world a global economic heart attack and took 600,000 lives in the United States. It's a very small life form, and there's very little of it, and it is doing incalculable damage to us. And it's by no means finished with. I think that it is not possible to conceive that we will be able to eradicate the coronavirus now, even with all the vaccines. COVID is going to be with us in our lifetimes for the foreseeable future. So what does the future hold in terms of these viruses? The coronavirus pandemic is, it's not an isolated event by any means. It's part of a pattern of events. And, we've seen, probably in the last generation, we've seen the emergence of these new infectious viruses that seem to be coming out of nowhere, that are in fact leaking out of the Earth's ecosystems, getting into humans, and then spreading. For example, HIV, Ebola virus, SARS virus, MERS virus, now COVID. And there are others. The names are not familiar to people, like Nipah, for example. So, are we going to see more of these pandemics in the future? And what is it going to be like? Well, I think that in order to see into the future, we can look into the past. And so I thought I would take a little bit of time here to talk about the great Ebola epidemic of 2014 and 15 in West Africa, which was the subject of my most recent book, to see what we can learn. Now, it began in December of 2013 in a tiny village in the nation of Guinea in West Africa. The village is called Meliandou. Here is Meliandou. It consists of about 30 houses, and there's a little clinic, and there's a school. And Meliandou sits at the foot of a deforested hill in an area of Guinea that used to be covered with tropical forest, but now has been largely denuded. This is happening all over West Africa. As the population increases, people cut down the trees, they sell the wood, and then they plant shifting agricultural fields in areas that used to be primeval forest. And as people do this, they come in contact with organisms in life, forms of life that people normally don't come into contact with. Now, in Meliandou, there was a dead tree. You can see photographs of it here. It was hollow, and inside the tree, there was a colony of bats. The bats are a particular kind that the local people called the flying mouse. Flying mice smell bad. They smell like mouse pee, and that's why the people called them flying mice. Now, people in that area like to eat bats, but they like fruit bats. Fruit bats are said to taste pretty good in a stew, but the adults don't like to eat these flying mice. They think they smell bad and taste bad, but kids will eat them. And the children of Meliandou like to gather around this dead tree. They would build a fire in the base of the tree. You can see some ashes there in that center image. And then they would let the smoke go up through the tree, which would knock out the bats, and they would fall down through the tree into the fire. The kids would stab the bats with sticks and then roast them on the fire and eat them like marshmallows. They would also share a bat on a stick with each other. There was a little kid, you can see him here on the left, named Emile Uamuno. He is there with his parents. He was playing around that tree one day, and we don't know what he did, but apparently he came into contact with one of these bats, a flying mouse. And not too long before Christmas, he developed black diarrhea, a high fever, and then he died in his mother's arms. Not too long after that, she developed a bloody nose, bleeding from her mouth, black diarrhea. She died. Emile's little sister died. Basically, everybody in the family died. It was Ebola virus. It wasn't recognized for a while. But when it finally was, by that time it was too late, and Ebola was circulating widely in villages and in small cities in West Africa. And over time, the virus replicated explosively in people. Now, Ebola virus is rather not much like COVID. Unlike the COVID virus, travels very readily through the air from person to person. Ebola virus is largely a direct contact virus. You get it when you have contact with blood and body fluids of somebody who's infected with Ebola. But if a single particle of Ebola gets into the human bloodstream, scientists have basically discovered that an infection by a single viable particle of Ebola can lead to a fatal infection, and you die with horrendous symptoms that include a complete destruction of the immune system, hemorrhaging from the openings of the body, and other things, including psychosis, and bruising in the body. It's a really, really horrible virus. So as it circulated and as it grew, Doctors Without Borders set up these camps, white tents, and they began, treating local people. And there really, at the time, there was no vaccine, and there was and really still is no effective medical treatment for somebody who is sick. But the local people really resented the Ebola doctors. And there was tremendous resistance to efforts to control the virus, to educate people about it. And what people said was a bit reminiscent of what we heard later in the United States about COVID. People were saying, well, but the virus doesn't exist. This is a political plot. This is a conspiracy. Also, local people were saying, well, it's not that bad. It's really not that bad. I don't really have to take precautions because, it's not going to get me. There was also a belief that it's over there, but it won't get here. And I think there was a tendency in the United States to kind of look down on Africans and say, oh, well, these people, they're not very well educated and, superstitious, and, it's a terrible thing to see them resisting the doctors and so forth and so on. But I would like to draw a comparison here. This is a house in Mission Hills, a really nice suburb of Kansas City. Now, what if, let's just say, some foreigners, a bunch of very wealthy, well-connected foreigners who had political power came to Mission Hills, and they had dark skin, and they spoke English with an accent, a really bad accent, or they didn't speak English at all. And they told everybody in Mission Hills that there was a virus among them, and that this virus had to be controlled. And the foreigners then set up tent camps with white tents. And they ordered the people of Mission Hills to go into those camps. And in Mission Hills, they noticed that if you went into one of those camps, you never came out again. And then people in Mission Hills began noticing that they were burying a lot of people right next to the camp, and that some of those white bags that were coming out of the camp were small and contained dead children. I think you can bet your bottom dollar that people in Mission Hills would be reaching for their guns and trying to get the hell out of Mission Hills as fast as they could. So, I think it's understandable what the local reaction was to Doctors Without Borders and the other medical agencies that came in there trying to help. Now, Crisis in the Red Zone, the story focused on one little forgotten hospital in a very forgotten place in the nation of Sierra Leone, the town of Kenema. Here we are at the public market to give you a sense of the flavor of Kenema. It's a really lively, interesting, very poor West African community. In that town, there is the Kenema Government Hospital. This is the one source of medical care that you can get if you're in Kenema. And the hospital itself got quickly overwhelmed with Ebola patients. The principal doctor there was Dr. Umar Khan. He was a scientist and a researcher, an expert on viruses, and who worked and collaborated with Americans here in the United States. Umar Khan was very familiar with the United States. Many members of his family live in the US. And he himself, his family members were urging him to come to the United States and go into practice in Philadelphia. And he told his family, they don't need me in Philadelphia. They need me here. So he stayed in Kenema. He stayed at the hospital as Ebola overwhelmed the place. He worked in a ward where it was a ward that was set up for biohazardous patients. It had a maximum of 12 beds. And in the end, there were 35 to 40 people in that ward. There were 2 and 3 people per bed. There were children and adults lying together in the same bed, people dying. And then they were so understaffed, and so many of the nurses themselves caught Ebola that they couldn't remove the bodies fast enough. It was a horrific situation at the Kenema hospital. And ultimately, Dr. Khan himself contracted Ebola and died of it. As he lay dying in a camp being run by Doctors Without Borders, there was an experimental drug, an antibody drug, that could have been given to him. And the doctors at the camp debated, should we offer him this drug? It had never been tried in a human being before. There was laboratory evidence to suggest that the drug might be able to save his life. But the whole question was, who is Dr. Khan? Is he a local African individual, in which case he would not be eligible for special treatment and special care? Or is he an international figure? The international doctors who were working in the Ebola outbreak had the option of being flown to Geneva and given this particular drug. They could have their choice of it. So the question was really, who is Dr. Khan? And it brought to the surface all of the history of colonialism in Africa, the question of, is Dr. Khan an equal to the international scientists? He's an international scientist himself. His collaborators are American. What is he entitled to? What should he be offered? And in the end, the managers of the Doctors Without Borders camp made a decision not to tell him about the drug and not to offer it to him. They did not know that he had privately told his colleagues that if the drug was available, he would certainly take it, but he was never told that it was available. Days later, the same dose of the drug that had been denied to Dr. Khan was flown to another location in Liberia and was administered to 2 Americans, medical workers, one of them an MD. And the doctor who got the drug, Dr. Kent Brantley, he was actively dying at the time the drug was given to him. He was minutes to hours from death. And the drug, which is called ZMapp, reversed the symptoms in Dr. Brantley in 90 minutes. And he got out of bed and he walked into the bathroom for the first time in 3 days. He had been hemorrhaging from all the openings of his body. The hemorrhaging stopped and he survived. There's no question that the drug saved his life. And when the medical people at the Doctors Without Borders camp found out that the drug had saved this white American, they were horrified and chagrined. And I think some of them realized that they had made a mistake, that their decision had been the wrong one. In any event, Dr. Khan was buried in the hospital where he served. And the hospital came through in the end all right. And Ebola in the end was beaten down in West Africa. It had the potential to become a global pandemic, but that never happened. And it's worth asking why. Turns out that, that the ordinary West Africans who worked in that hospital, some ran away from the fight, but a handful stayed. And they threw themselves into the fight the way Dr. Khan did. They knew they were going to die, and they went into situations in wards where it was inevitable if they were going to be infected. And they did die. And in the hospital, in the end, lost 37 nurses and physicians, as well as hundreds of patients. But they managed to get things under control. And in a larger sense, the same thing happened all over West Africa. Eventually, people woke up and their illusions about Ebola went away. And I think it is safe to say that these ordinary Africans saved us. They stopped Ebola. And they did it in the old-fashioned way. They learned the signs. They learned how to recognize a patient with Ebola, which was something that American doctors could not do. They practiced harsh social distancing, incredibly harsh. If one member of a family was diagnosed with Ebola, the entire family would be shunned. People who were in the street dying of Ebola, nobody would go near them. They died alone in the street. People became compulsive hand washing fanatics with chlorine. And finally, in the villages, in the rural areas, the villages went back to a traditional way of controlling infectious disease in Africa, which is what is called reverse quarantine, where you close yourself off to the outside world. These villages shut themselves down and they wouldn't let anybody come in. Nobody could go out, nobody could come in. That is reverse quarantine. And they stopped the virus. Some of these people who worked in the Ebola outbreak remind me of soldiers in the American Civil War, going into battles like Antietam, or in World War I, where they knew they were going to die, but they went and they did their duty anyway. Here's a man, Joseph Henry Moseret. He's a hospital janitor. I know him. I respect him greatly. He earns maybe $3 a day, maybe, cleaning toilets. And he cleaned toilets, Ebola toilets, during the outbreak. At first, he had spacesuit he could wear, but they ran out of biohazard gear. So he dispensed with biohazard gear and he just went on cleaning the toilets without protection. He survived. He didn't get Ebola. And it was people like him, many, many people like him, that went into the battle and they stopped this enemy of the human species. And one of the doctors said to me, outbreaks end when people decide they will end, that you end an outbreak with behavior. And right now in the United States, I think it's fair to say that a very large fraction of the American people has still not decided that they want the COVID outbreak to end. They're resisting masks and resisting vaccination. Maybe they will change their mind or maybe they won't, I don't know. But I know that the Ebola outbreak stopped when Africans decided they'd had enough. Now, I want to tackle the whole question of did the COVID virus come from a lab? The early cases were found connected with a wet market. That's a market where animal products are sold, especially wild animals, in Wuhan, in the city of Wuhan. The early cases came from around there. Now, researchers, when they were investigating these bats that carry coronavirus, they went into a lot of different caves in Yunnan. They collected these many, many samples. They identified 500 new, previously unknown coronaviruses. And they estimated that there could be as many as 5,000 different coronaviruses circulating in bats in Yunnan, which is about 700 miles from the city of Wuhan. But they also found that people in these villages like to eat bats. And they began testing, the researchers began testing the blood of villagers near caves. And they found that a significant fraction of the villagers in each of these villages, maybe 10, 15%, had antibodies to a coronavirus, some coronavirus, not COVID, not COVID-19 virus, but some virus, the corona. And what it looks like is that people are constantly coming into contact with bats, perhaps through cuisine or who knows how, but that there's a lot of coronavirus in these villages. People may be dying of coronaviruses, or they may be getting, something like a sniffle, just like a bat sniffle. We don't really know. But we do know that there is a continual spillover of coronaviruses into the human population of Western China. It's just going on constantly, and it's been going on from time immemorial. An American researcher, Peter Daszak, is one of the individuals who went into these caves and ran these expeditions. And he has been closely associated also with the Wuhan Institute of Virology. Now, this is a biosafety level 4 lab where coronaviruses are studied by Dr. Shi Zhengli. You see here, she's in a spacesuit. She's an extremely well-regarded, admired researcher, internationally respected, who was one of the key figures who discovered the origin of the original SARS virus. She traced it to bats. And she speaks fluent English. She's very well liked by her American colleagues who work with her, including Peter Daszak. And, you know, for a long time, you know, when speculation came about that possibly the COVID virus had escaped from Dr. Shi's laboratory, or that maybe somebody had made it in Dr. Shi's laboratory. Dr. Shi herself was eloquent. She said, you know, that she had panicked when she first learned of the outbreak of COVID, and she had gone through all of her laboratory collections and samples, trying to determine whether she had a virus in her lab that was like the one that was beginning to circulate in Wuhan. And she didn't find it. And I think she was quite convincing with people. She seemed utterly sincere about this. She said, I was terrified. I went through everything. I looked as carefully as I could. We do not have that virus in our laboratory. Now, there is another virology laboratory in Wuhan, and they have not shared their information, their data, or their records. So it's always possible that the virus could have come from there. But did it maybe come from Dr. Shi's lab anyway? Is she covering something up? We don't know, but here's the case for the possibility of an escape. Now, what I think, the bottom line is, I think that there are two hypotheses that are both untested. One hypothesis is that it emerged from nature, that it went from a bat to possibly an intermediate animal, and then somehow or other it got into a human, perhaps at the wet market in Wuhan. Another hypothesis that also needs to be tested is, could it have emerged from Dr. Shi's lab through an accident? And you know, there have been some indications that you know, that things are maybe not perfect at Dr. Shi's lab. One is that there has been this report through American intelligence sources that 3 individuals in her lab in December 2020 got sick, really sick, with respiratory illness and had to go to the hospital. Dr. Shi denies this angrily. Scientists have also looked very carefully at the genetic code of the virus. Now, if it was a weapon, or if it had been engineered in a laboratory, there would be telltale signs in the RNA of the virus, in the genetic code of the virus. You might see foreign genes that don't normally belong in a coronavirus in there. You might see sequences that are called promoter sequences that would be introduced through a laboratory procedure, but nothing like that has turned up in the genetic information of the virus in the genome. Is it a weapon? Well, if it's not a weapon, in my view, it can't possibly be a weapon because it's not that good. And believe me, there are bioweaponeers in the world today. There are nations that are almost certainly developing and working on biological weapons, including Russia. And you know, they have all the tools of modern biology at their disposal. And if they wanted to make a weapon, it would be a really good weapon and it would be well engineered. But COVID is not well engineered in that sense at all. Now, it is also the case that Dr. Shi and her colleagues were doing experiments at their lab known as gain of function experiments. Now, in these experiments, researchers take pieces of coronavirus, they graft them onto pieces of other viruses, and then they try to determine how evolution might proceed to make a coronavirus more deadly and more infectious in humans, to try to predict how the virus might evolve over time. These gain of function types of experiments have been extremely controversial in the scientific community. And there are those, I am among them, who feel that the gain of function experiments with viruses are inherently risky and should not be done unless there is a compelling, overwhelming reason to do them. But Dr. Shi was doing them. And I am not sure exactly what she was doing or what kinds of pieces of viruses she was using. Some of these gain of function experiments were being done under less than total biological safety. The experiments were being done at biosafety level 2, which is basically very little biosafety at all. And could possibly a gain of function experiment have on a bat coronavirus ended up with COVID-19? The jury is out on that. We don't know for sure. But then at the risk of getting a little too technical, there is in the genome of the virus, there is something called the furan cleft. And the furan cleft is a stretch of code that codes for the exact shape of the protein, the little sticky blob of protein on the outside of the virus, those little blob-like hairs on the outside of the virus. They're used to attach to human cells, and then the virus uses them to get inside the cell, to invade a human cell. And it's been known for a long time to scientists that if you want to make a virus more infectious in human cells, you can put the code for this thing called the furan cleft into the knobs on the outside of the virus, and that will enable the virus to much more easily enter a human cell. So the COVID-19 virus does in fact have a furan cleft in its code, and no other coronavirus from bats that we know of has the furan cleft. So did it get there through a gain of function experiment that they were doing that got out of hand? We do not know. But the Chinese government has refused to share any data or information or records from the Wuhan labs. And they also have refused to allow international teams to go in and do solid on-the-ground epidemiology to try to trace the virus back to the first people who had it. China is stonewalling. But David Baltimore, a Nobel Prize-winning microbiologist and molecular biologist, who later was the president of Caltech, California Institute of Technology, when he heard about the furan cleft in the code of the COVID-19 virus, he said, that does it for me. It convinced him that the virus was a laboratory escape. Now, David Baltimore is a very smart man, and he's also conservative. He's not a wild person by any means. He's very, very solid. But when he said that, I think a lot of people kind of had their eyes opened. But we still don't know. And it's also possible, it's very possible, since there are thousands of coronaviruses out there, as far as we know, that it could be very, very possible that one of them had the furan cleft in it naturally. And in fact, that's why it has been so successful as it's moved into the human species, because by just a matter of chance, it already had that particular little bit of genetic code that enabled it to get into human cells very easily. So will we ever know whether it came from a lab or whether it was a natural outbreak? The only way we're ever going to know is by old-fashioned, classic shoe leather epidemiology. There should be international teams that should go into China and they should do the work. Interview people carefully, review lab records, make sure that they see all the lab records. But whether that can actually happen, whether the Chinese government will ever permit that, is anybody's guess. I suspect that they might not. And I wonder sometimes if the reason for the Chinese clampdown and secrecy is because they themselves don't know where the virus came from. But they would be incredibly embarrassed to admit to the world that they don't know, that their epidemiology itself isn't good enough to find out. So at any rate, while that's a mystery, we do have one incredible success, which is the development of these vaccines. It is a miracle of science. And the heroism and the dedication and the incredible ingenuity that has led to the development of highly effective vaccines in a very short period of time is something that we can all celebrate. It's a really incredible achievement, and it bodes well in our future relationship with the virus sphere, with viruses continually jumping species and occasionally landing in us. I wanted to leave you with one final thing. When I was in Kenema, the markets had been closed and Ebola was still in the area and people were still dying of Ebola. And I was there as a reporter trying to get the story. And they opened up the market for the first time in a long time, in a year, almost a year. And I went in to the city market and I began walking around and talking to people. And I had a camera with me. And the older people, a lot of them were saying they didn't want their picture taken. They were saying, no snap, meaning don't take my picture. But there were these kids. There's this young lady, and she came right up to me and she said, take my snap. And so I did. And I think this girl is fantastic. She's looking you right in the eye. Her friends are kind of giggling and laughing. And she is filled with self-confidence. This girl has the world in the palm of her hand. And this is, don't forget, it's right in the middle of an Ebola outbreak. But there she is, and she is the future of Africa and the future of our world. And she is what public health is all about. This is a young lady who has dreams. I don't know where she is right now, and I don't know what she's doing, but I have hope for her, and I have hope for her generation. And I think when we think about what public health doctors do, and what the scientists have done in developing this vaccine, and what we have going for us that viruses don't have, we have the ability to work in teams. We have consciousness, awareness. We have the ability to self-sacrifice, to sacrifice our life to help someone else's life. And we also have the ability to change. Viruses can change, but we can change faster than they can. And I think in the long run, we're going to be okay. And we're going to be okay for the sake of this young lady. And she's going to be one of the people who helps us be okay. Thank you very much.
Richard, thanks so much. We've got a number of questions here. If we can take maybe 10 minutes to try to get as many answered as we can. And I just want to start the perhaps telltale Furan cleft that you talked about. Do we have any idea if it has been unique to this virus? Was science evolved enough in previous pandemics to be able to identify that?
That's a really interesting question. I hope I know enough to be able to be knowledgeable to answer your question. But the Furan cleft has been known to molecular biologists for quite some time. And it is common in viruses that use these glycoproteins, these sticky little knobs, to attach to human cells and enter human cells, get into the cells. So it's a piece of code that is apparently, fairly, not uncommon in the viral world, in the viral kingdoms, naturally. But it's also something that molecular biologists have, they know the code. And, nowadays there are machines where you basically, they're like a typewriter. You can type a genetic code into the machine and it will produce a length of DNA that has that code. And then you can take that DNA and you graft it into an organism. But to address your question, I'm really not sure how long it's been in terms of how long has the scientific community actually known about the fern cleft. I'm not really sure about that.
Audience question, addressing the wet markets, which we've heard about in cases even prior to this being problematic. Do you get a sense that there's any sentiment in China or even an ability in China to outlaw those markets, to perhaps eliminate that threat? And are there enough alternative food sources to be able to do that?
Yeah, this is a great question. Even though China is an authoritarian state, I don't think they have the political power or the will to completely eliminate the wet markets. The wet markets they've been a part of Chinese culture for, as far as we know, thousands of years. And the eating of wild creatures, it's gourmet cuisine is what it really is. And we have the same thing in the United States. Lobsters, caviar caught from wild fish, wild caught salmon, for example. But in China, just as here in the United States, we have salmon farms. In China, there are these large farms where wild animals are grown for human consumption. And so you think about a wet market and you think, well, the animals that are being sold there for food were all caught in the wild. Well, not so. They were actually raised on farms, a lot of them. And there are many such farms around Wuhan, the city. So some people have said, well, how did that virus get 700 miles from the caves in Yunnan all the way to Wuhan? It must have been brought there by, in the body of a researcher who'd been in a cave and then they got sick and they brought it back to Wuhan, But it's actually, on Occam's, the principle of Occam's razor, the simplest explanation you could look for first. A simpler explanation is that there are these wild animal farms all around Wuhan, and the virus actually got into one of those farms and then got into the human population that way. But the question, could this be eliminated? I don't think so. I don't think so.
Question again from the audience, asking if they found the COVID-19 virus in any animal yet. And if I recall, they have been found in some zoo animals. But further question was, has it been found in the wild? Yeah, no, it has not. And that's one of the items that's contributing to this debate, these questions about whether it came from a lab. Now, a virus has been identified that's a fairly close relative of SARS coronavirus 2, the COVID-19 virus. It's a bat virus that was recovered from a bat from a cave, and it has about 96% similarity to the code of the human COVID virus. But a 96% similarity is really not that similar. So one of the things that I would love to see the epidemiologists do is do that legwork and identify an actual natural animal that the virus has been found in. Right? That's pretty difficult to do. If there are 5,000 different coronaviruses out there in caves all over the place, man, you're going to have to collect a lot of bat blood
before you might find, the COVID-19 virus.
Can you speak to the importance of identifying patient zero?
Well, it's incredibly important. With Ebola outbreaks, it's been extremely difficult to identify patient zero, because at that fracture point, that point where Ebola breaks into the human species, gets into one person, but then it's so lethal that typically all the early cases of it, they're all dead. So there's nobody to interview. All you can do is talk to the relatives of the survivors. And, it's like going into a place where a bomb went off and trying to reconstruct what happened. But with COVID, it's a little different. Presumably, early cases easily could have died, but also they could have survived. And if you go and you really do your stuff, you start interviewing people, you start trying to trace chains of infection, you might just be able to locate, if not the very first person, you might be able to get close to them or closer to them.
What have we learned, do you think, in the past 15 months that should help managing the next pandemic if properly applied? And that's kind of an expansion on an audience question about whether you sense that people in America have learned the lessons that the West Africans learned with Ebola, including the fact that, politicization is probably, one of the worst things for public health.
Yeah, well, I think we've learned the lesson imperfectly. I would hope over time we would learn it better. I don't really know if we will. But there are other very important lessons that we've learned. One of them is that public health is a matter of national security. And any money spent on the CDC and on state and even local health departments has a huge payoff. If we had been able to identify COVID early enough, think how many billions and trillions of dollars in global economy could have been saved. Think of how many lives could have been saved. What's that worth to the global economy? It is, I can't state this enough. Public health is just supremely important for national security. And what that means is that a crisis in a rural hospital in West Africa is a crisis for the United States, because those cases in that little hospital could have come to the United States. The virus could have gotten here. And so, there are things we can do. Just basic public health measures, things like having enough surveillance teams out in the field all over the world, just watching and looking for emergences of unexplained new diseases. That can be incredibly important for stopping or slowing down a potential pandemic. The next thing that I think we've learned is our government and pharmaceutical companies have learned how to work fast and work together to produce vaccines quickly, to get potential vaccines out there and then to get them into the testing pipeline as fast as possible. Now, I know that at Moderna, which developed one of the vaccines, the company they heard about this outbreak in China, and they were able to get the genetic code for the virus from the Chinese researcher. And I think by 10 days later, they had already developed a vaccine ready for testing. This was even before this was known to be a public health emergency, but they were on it. And so what took the time was not actually the invention of the Moderna vaccine, the Moderna vaccine, I beg your pardon, but testing it, determining that it was efficacious and safe.
As we wrap up, there will no doubt be books, probably many books written about this pandemic. Is there one formulating in your mind?
I think not, actually. I'm at work on another project, and I thought, I've written a lot about viruses. I'm not sure I have it in me to write more.
Well, as I said at the outset, I think you can bank on hearing from us again. We'll look forward to that book coming out. You can just probably count on a call from us. This was terrific tonight. It was timely. I have a whole sheet full of additional questions I would love to ask you. I could go on another hour, but this has been tremendous insight. It has been just exactly, what I expected. You, I do think, more than perhaps anybody else can properly put all of this into context. So thank you so much for being with us tonight.
Well, it's been great to be with you.
And everybody out there, thank you for joining us. We'll look forward to seeing you again. Thanks again, Richard.